Super.
And it appears that we are officially live. Hello, everybody, and welcome to another session of our Sussex Vision seminar series, as always within the Worldwide Neuro Initiative. I'm George Kafetsis, a master's graduate from Thomas Euler's lab and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from the Leibniz Institute for Evolution and Biodiversity and the Museum für Naturkunde, Dr. Lauren Sumner-Rooney. Similar to today's topic, Lauren's steps are tales of vision in different invertebrates. Following her bachelor's degree in biological sciences from Oxford University, Lauren joined Julia Sigwart at Queen's University in Belfast for her PhD in sensory systems in marine invertebrates with a focus on mollusks and echinoderms. She then moved to Germany and Berlin and Museum für Naturkunde as a Leibniz DAAD research fellow, where she studied vision in brittle stars before returning to Oxford in 2017 to investigate evolution of vision in spiders. Late last year, she returned to the Museum für Naturkunde in Berlin, which is Museum for Natural Sciences, in case you don't speak German, this time as a junior group leader, starting her lab and aptly naming it Multiply. It is a great pleasure to have Lauren today with us, talking about relatively under or completely unexplored visual systems. And surely together with many people in the audience, I cannot wait to see how they differ or resemble those in traditionally studied model groups and why some animals have more than two eyes. So without any further ado from my side, please all welcome Dr. Sumner-Rooney. Lauren, the stage is officially all yours.
Brilliant. Thank you so much for the introduction and of course for the invitation here today as well. I'm very excited to be talking to you all. And good morning, afternoon or evening, depending on where you are. So as George said, I'm actually going to be talking a bit today about some work that has been ongoing for a while, but has kind of come to a head in the last kind of few months since I moved to Germany to start my own group. So obviously I'm preaching to the choir when I say that vision is a really important sense. So light is obviously hugely important in biology and it affects all sorts of different processes. It's very widely appreciated to be a critical biological innovation. So not only things like foraging, mate finding, the assessment of avoidance of predators and navigation, vision and light have really far-reaching impacts for all sorts of aspects of ecology and evolution. And one of the results of this is this amazing...
Lauren, sorry for interrupting you. I don't know, would you like to start screen sharing or this is pre-mode?
I will start screen sharing. Thank you for checking. But you can all imagine a beautiful diversity of eyes that you can see in front of you right now. So let me just try that again. Thank you, George. All right, take two. Hopefully you can now see aforementioned beautiful diversity of eyes. Good. So as I said, we actually have this really stunning diversity of forms that have evolved famously by Mayer and Salvini-Plowman's estimate more than 40 times across animal diversity. Now, this is obviously a very well-established research field, vision in general, but also it has impacts on behavioral biology, ecology, and of course, bio-inspired design as well. And this is actually a tradition that stretches back centuries. So being very visual creatures ourselves, actually researchers have been fascinated by vision in both humans and in animals for a very, very long time. So you can see some of these illustrations from the 17th and 18th centuries really nicely depicting not only human eyes, but also those of insects as well. But the majority of our efforts have actually kind of focused on one particular type of vision, and that is binocular vision. Obviously, this is the most familiar to us, but it's actually dominated the field for centuries. Obviously, that means that we understand the advantages of binocularity pretty well now. So from stereoscopy and depth perception through to having an expanded field of view, and of course, redundancy in having two eyes as opposed to putting all your eggs in one basket, as it were. But it's by no means the only strategy out there. So having many eyes or multiocularity does occur across metazoa. We've only really been studying a tiny fraction of the existing diversity. Now, obviously, this isn't new. This is not something I've discovered. We've been aware of a lot of these systems for decades or even centuries, or in many cases have at least suspected a visual function of some of the structures that we're going to see today. In a small number of cases, we actually have a pretty good understanding of how they work. So one example in particular would be scallop eyes, which were first described in 1795. They're very distinctive, and of course, they're in an animal that's accessible and delicious, so we know quite a lot about them. We also know a lot about the structure of individual eyes, including some visually guided behaviors. And actually, we're now starting to have a discussion about how integration occurs between eyes and systems like this. This is very challenging. Obviously, the more eyes you have, the more units you're integrating, the more difficult this is. But we are starting to see some really nice experimental work looking at this. So in the example of scallops, there was a great paper from Dan Chappell at the end of last year, looking at how different eyes might be communicating and the difference between spatial vision and spatial resolution. So although we've known about a lot of these systems kind of in isolation, or we've been studying them in isolation, actually the kind of use of phrases like distributed vision or dispersed vision are actually relatively recent. So in the last kind of 10, 20 years, more and more conversation has been happening about what these systems are overall. This wider conversation about why they evolve. And this is basically what I'm hoping to address along with several other people in our new group in Berlin. So although for specific questions, we're inevitably taking a model-based approach, what we do want is have a kind of broader conversation about conceptual questions and links between all of these very different systems. So really starting at the top, we want to answer some pretty basic questions about some of these things actually, and you'd be surprised that we don't necessarily have the answers immediately accessible to some of these questions. So for example, where do these actually occur? I struggled to find a kind of nice, neat list of all of the phyla in which you can find these many-eyed visual systems. And are there kind of unifying features? Are there functional principles that kind of govern the relationships between different characters of these systems? And are there specific ecological or developmental correlates, as there are for lots of other aspects of binocular systems, for example? So the first thing to do, as I said, was actually just to kind of try and survey the animal kingdom in its broadest sense and establish exactly where and when these visual systems appear. So what we did was actually, and this is ongoing, what we did was look at all phyla that have more than 1,000 described species. Obviously, a lot of these structures have not been tested specifically to work out whether they are in fact eyes in the sense that they convey spatial resolution. So we've taken quite a generous approach to this and included any structure that has more than 2 photoreceptors and it has squalene pigment. So as a kind of very, very basic possibility of discerning which direction that light is coming from, and we're looking at adult structures only. So one of the things that we've done is try and then extract data about all of those different visual systems, the number of eyes, the number of eye types, their diameter, body size, and a couple of basics to do with photoreceptor length or interoceptor angle and diameter. So what we found is that this common perception of many-eyed systems being peripheral or unrepresentative is actually quite a misrepresentation. So these systems actually occur in 7 out of the 10 largest animal phyla, and the remaining 3 of those don't have eyes at all as adults. So actually they're pretty ubiquitous amongst any phylum that has eyes in any case. They're also in at least 9 out of the 16 phyla that contain more than 1,000 species, and they obviously also appear in the fossil record more than 500 million years ago. So these are not unusual, even though they seem alien to us. They're actually a very common solution to the evolutionary problem of vision. One of the things that has actually kind of emerged from this, and I'm not going to give you a rundown of all of these systems, we'd be here all night, but what has kind of emerged is these 3 broad classifications that we kind of propose adopting in future conversations about many-eyed systems. So first of all, you have duplicated systems. So this is where you have many replicated units that have shared developmental origins and collect broadly similar or the same information. So examples might include the scallop that we already discussed, perhaps chitin shell eyes, and the optic cushions of sea stars as well. This is just where you have a single type of visual system, but perhaps it's multiplied or spread across the entire body. The second type is parallel visual systems. So this is where I'm kind of talking about having more than one type of eye with distinct developmental origins and presumably collecting different visual information. So the best known of these, of course, would be the insect ocelli and compound eyes. But then you also have things like the box jellyfish and their 6 eye types on their ropalia, as well as vertebrate parietal eyes, horseshoe crabs, etc. And finally, we have this somewhat kind of enigmatic category, which you could argue, does it fit into the kind of whole concept of being a many-eyed system? What we have here is extraocular systems. So wherein an animal lacks a discrete visual structure, a kind of dedicated organ, and actually has photoreceptors spread across the whole body. This obviously in itself occurs in quite a lot of different systems, but we only have direct experimental evidence for spatial resolution from those systems in a couple of different species. So for now, it's only been described in a couple of echinoderms, but it's highly likely that there may be other groups as well that could eventually fall into this category. So they're both very widespread, and they also appear to be have multiple origins as well. So the numbers in parentheses here are just estimates of how many potential origins there probably are, according to the position of these different systems in animal phylogeny. So what about these functional and ecological relationships then? So we started to have a look at some of those data I talked about, things like eye number, eye diameter, but it's, as I said, an ongoing process, so do bear with me. These are kind of first pass visualizations to get an idea of what's happening in the data, and hopefully this will be published more formally as a review later this year. We do have additional phyla to add still, and then we can perform proper phylogenetically corrected models, but I do think there are some interesting leads, and I just wanted to kind of share them with you and also get your thoughts as well. So by functional principles, I actually kind of mean basic relationships between different characters of the visual system. So within single eyes, for example, we know that there are often trade-offs between resolution, sensitivity, and eye size, among many, many other factors. I suppose comparably, if you're thinking about binocular systems, we know that the spectrum of kind of field of view coverage versus depth perception can have links to trophic level. So in these many-eyed systems, there are a few new variables to consider here, I suppose. So we might expect to see relationships between eye number, eye size, number of eye types, and of course, there are going to be evolutionary factors or constraints and ecological factors to include as well. So as I said, we're kind of still gathering data on this, but so far we've got 126 species from 8 different phyla, with kind of more being added all the time. So we've started to get a handle on some of these relationships, and they're kind of very basic form. So for example, unsurprisingly perhaps, eye number and eye diameter have show quite a strong inverse relationship. I mean, this is perfectly logical. You can imagine that you'd make a greater investment in fewer eyes or less investment in more eyes. This holds for both absolute eye size, which of course is functionally important to things like sensitivity and resolution, and of course to relative eye diameter as well, so adjusted for body size. Obviously, as I said, it's important to note that this doesn't include phylogeny in the model yet, but actually if you unpick it by phylum, this relationship broadly carries, and actually some of those phyla do have quite a nice coverage of eye number as well. So as an example, again, this is not necessarily new knowledge, but this slide of Mike Box really nicely demonstrates this concept in some of his surveillance. So this idea of either having distributed eyes, which often be much smaller and spread across a larger part of the body, versus consolidating all of your investment into 2 much larger, possibly more powerful eyes there. But if we see this across multiple phyla, then perhaps it's a kind of broader principle of these duplicated systems anyway. So the next thing that we actually looked at was whether or not body size affects any of these things. So what we found was that parallel systems are actually more common in smaller taxa. So again, this makes sense. It seems like a fairly logical result. So if you imagine a smaller animal might find it more energetically economical to split tasks between several smaller eyes rather than having fewer large eyes. So I guess doubling investment by adding an extra eye of the same size might be more economical than increasing the volume of an eye by a factor of 2. And actually, there's quite a nice analogy on this from. Oh, I think I might have frozen. I hope not. Can you still hear me?
Yeah, we can hear you crystal clear.
Okay. Good, fine. You froze, so I was worried that I might have dropped out of my internet there. So actually, in some of the best targeting advertising I've received in my entire life, Samsung have actually really nicely summarized this concept. So what we now see increasingly in smartphones, for example, is that actually you can't buy something that has fewer than 4 cameras, but all 4 of those cameras are adjusted to do a slightly different job. So when you have these kind of size constraints and space constraints, it might be more economical to produce more than one eye type than it is to make a larger eye that can do more. So potentially tied to this with a kind of eye size, eye number relationship, we also see fewer eyes overall where you have more types of eyes, so in parallel systems. This is probably going to be affected once more vertebrate data are added, but it does seem to, at least seems that our eye number becomes less variable as you increase the number of parallel systems. So if we're looking at ecological factors, activity and trophic level both seem to affect eye number, and of course they're almost certainly linked to one another as well. So sessile species and those that are primary consumers seem to, generally speaking, have more eyes than either their more mobile or their higher trophic level counterparts. We didn't see any impact of things like body symmetry. We did see a difference between having more eyes in aquatic than terrestrial taxa, but of course there's much greater representation, much greater phylogenetic representation in aquatic taxa. So that's a bit of a confounding factor there. If you look at eye diameter, then again, kind of closely linked, I suppose, we actually do see the opposite effect. So those taxa that move in 3 dimensions or moving more quickly have substantially larger eyes than their counterparts, and again, terrestrial animals do as well. Finally, looking at the links between, for example, ecological factors and the number of eye types, this requires a bit more work, a bit more data, but we do seem to see a couple of patterns. So, for example, sessile and slow-moving animals are more likely to have just 1 or 2 eye types, and the same for both locomotion and speed seem to affect the number of eye types that could be present. So from all of these things, we kind of have this spectrum emerging, perhaps, to consider for many eye systems. So things really stretching from having many small eyes of 1 or a few types, all the way through to having fewer larger eyes of different types collecting different information. And there do seem to be pretty fairly logical and fairly strong correlations there with both trophic level and with activity level as well. Obviously, again, there's a kind of phylogenetic element here, so we do have something of an overrepresentation of arthropods and vertebrates where you have parietal eyes at one end and the marine invertebrate taxa at the other end. So that's something that requires unpicking as well. And the 3 categories that we've kind of identified earlier also kind of broadly follow along the spectrum. Obviously, there's some overlap. There are plenty of examples of species that have both duplicated and parallel visual systems, so they kind of fall into this grey area in the middle. So today, one of the things that I want to do next is actually look a bit more closely at some of these factors, so eye size, eye function, and ecology and evolution. So to tackle more specific questions of exactly how these systems function and evolve, it obviously helps to constrain yourself to 1 or a few groups. So for me, spiders are really the kind of a really great model for this. So they occupy a spread on this kind of theoretical spectrum that we've spoken about, but they also exhibit a range of different visual ecologies. They have both parallel and duplicated systems, and they have a very constrained blueprint to their kind of anatomy. So what I'll talk about next is how then spiders have exploited this ability to use more than 2 eyes. This is where I warn you that you're about to see a lot of close-up pictures of spiders. Anybody who's not particularly a fan of that, I will flag up when they're gone so you can look back at the computer screen. But particularly when I was doing in-person seminars, I found that this warning became very helpful. So without further ado, this is some of the beautiful diversity that we see in spider visual systems. So as I said, they've got an enormous variety in size, arrangement, and of course, ecology as well. So you can see that there's a multitude of different evolutionary routes that spiders have taken to having this quite constrained visual system. The spider photos are now mostly gone. But as I said, they all use quite a conserved blueprint. So as a quick primer, I have this beautiful illustration of named warehouses, which I can't improve upon, so I'll just use it. So they have 2 types of eyes and usually have 4 pairs. They have the principal eyes with an inverse retina, and the secondary eyes usually with a tapetum and the inverse retina on the right-hand side there. And we do see a kind of really beautiful array of different arrangements, but we also see different visual ecologies. So for our visual hunters, free-running hunters, and of course web-based hunters, we do see some kind of like very superficial correlations with their visual system structure as well. So the first challenge is obviously understanding what it is that they're actually seeing. So what information every animal collects, what information every pair of eyes collects, but only a very small number of species have been studied in great detail so far. Then I'm kind of interested in testing some of the principles that we identified in that kind of like broader survey. Do they hold up if you're just looking at a constrained system, both in terms of eye number and in terms of phylogeny? And otherwise, how much of that spectrum is actually just a phylogenetic artifact? And then finally, what are the kind of nuts and bolts of changing these visual systems so dramatically? So obviously, thanks to more than a century of really excellent vision research on binocular animals, we have a really great understanding of some of the relationships between morphology and function. Obviously, in animal vision, these are quite closely related as a combination of optical and biological constraints, which is great. I mean, it means for us that we can learn quite a lot from structure, and actually we can kind of make inferences about 4 out of 5 of the kind of major functional properties of the visual system. We obviously can't make any inferences about wavelength sensitivity from structure alone. You need to have a little bit, well, unless you're using filters, I suppose, but we can certainly have a kind of good level of understanding, at least a comparative sense, of those other 4 qualities. So for some of these structural studies, we've used a technique called synchrotron tomography, which some of you will be familiar with. It's very similar to micro CT. It's much more powerful, so the scans are much faster. It uses more specific X-ray energies as well, so you can end up with better resolution and greater contrast enhancement. So just to give you an idea of what we produce, hopefully you'll be able to see a scan of a wolf spider right now, and you can see that you're passing through some of the eye pairs there appearing in the middle. So given that that total field of view is only a few millimeters across, we've got really beautiful high power, high resolution scans and a lot of information there, with each scan only taking about 8 minutes. So if we zoom into one of the particular eyes here, what you'll hopefully be able to see, I realize now that you probably can't see my cursor, but hopefully you can see my cursor, wonderful. Hopefully down here you can see this kind of beautiful striky region where we've actually got the individual rhabdoms here that you can see passing between the pigment here. So actually you can make out the rhabdoms of individual photoreceptors, and you'll notice that at this edge they're pretty wide, and in the middle they're very fine. So we're actually starting to see differences in the retinal mosaic just from these kind of raw scan data, which is really exciting. So again, many could be familiar with these techniques, but we're using these data then to generate 3D models, so we can actually look at the lens and the retina, particularly in their full kind of 3D context. So what we can do then is extract a huge amount of information about each eye, each species, and as I said, at least have a decent inference as to function in a comparative sense. So looking at comparing eyes and comparing species. So for example, here we can see that these 2 eyes, the posterior median and posterior lateral, may well be collecting similar information. So they've got small interoceptor angles, long rhabdoms, and a fairly high proportion of lens diameter to thickness for greater focusing power. It's likely that compared to the other 2 pairs, these eyes are more built for detail. And it's important to note here that they do also point in different directions. By contrast, if you look at the principal eyes, the AMEs, they're much smaller. Their field of view overlaps pretty much completely with the posterior median eyes, but they look like they're probably collecting different information. So here we have a much larger interoceptor angle, probably less focusing power, but we do have potential polarization sensitivity in this kind of tiered retina, where the two layers are oriented perpendicular to one another. So again, they're sampling perhaps the same space as the posterior median eyes, but they're extracting different information. So you can actually extract a lot of information from these measurements, but getting good data out of these retinal characters can be very, very slow. So a big part of my group's work is going to be moving towards semi-automated analysis of retinal mosaic structure. So thanks to collaborators like Dr. Pablo Correa at UCLA, he's actually developing a pipeline to extract much more detailed information from these reconstructed scans. So for example, this is just one eye of a crab spider, but you can see just from looking at these kind of retinal maps that you have clear regional differences in eye structure, as we saw from that example scan that I showed you. So for example, we have this kind of mid-band of smaller cross-sectional area of the rhabdoms, lower skewness, and a smaller interoceptor angle. So presumably, again, that's kind of like across the horizon of the eye, and that's also at the point where the retina is furthest from the lens and probably has, again, the best focus. But for now, we're working with our data being extracted manually, but we can still see that there's quite a different range of possible visual abilities, and that different eye pairs do behave differently in different species. So for example, there's a tendency for the anterior median eyes to have the finest potential resolution, And also we're kind of seeing outliers in a lot of these characteristics of visual hunters. So these two are highlighting jumping spiders and wolf spiders. And you can see that they actually kind of are sitting quite a long way outside the normal distributions of other spider families that are less reliant on vision. So this kind of increased variation between eye pairs relating to ecology is something that, again, was kind of supported by that original survey. So if we look at an example, so interrhabdomeric angle versus rhabdom length, then again, we can see how much variation there is between eye pairs in these kind of different ecological groups. So our web hunters show relatively little functional divergence between eye pairs here. You can actually see the kind of 3 secondary eye pairs kind of clustered together and a little bit of difference from the principal eyes here. But if you actually then start looking at, for example, free hunters and certainly visual hunters, there's much kind of a greater spread within individual animals across these different axes. And of course, as I said earlier, phylogeny is a really important factor here as well. So in spiders, there's a substantial phylogenetic element to hunting modes. Thanks to a fairly good understanding of spider phylogeny at the family level, we can then use that to reconstruct the evolution of some of these retinal characters in the different eye pairs. So for example, if we're looking at the possible polarization sensitivity, this perpendicular arrangement of the rhabdoms, we can see that it has occurred multiple times in different spider families and in different eye pairs, but it's overwhelmingly appearing after the kind of loss of aerial web-based hunting. So we do see kind of several different origins, but there may well be an ecological correlate there as well. This seems very sensible. Polarization sensitivity is often used for navigation in all sorts of different arthropods, particularly understood in insects. But in some spiders that are free hunters, particularly nephosids and zodariids here, some of those species will actually build themselves a little retreat, and they will try and return to that retreat once they're done foraging. So an extra navigational cue seems like a very sensible adaptation. Same for if you look at high resolution, here defined as kind of an interreceptor angle of less than 3 degrees. Again, we see multiple origins of this and in many different eye pairs. One thing that's interesting to note, of course, is that they're in different eye pairs from our polarization sensitivity. Once again, if you have small eyes, they probably can't do 2 different jobs. So 2 jobs in 2 different eye pairs. Even though it's obviously largely restricted to a single clade, so this RTA clade down here is where a lot of visual hunting occurs, there do seem to be multiple origins within that. So obviously not everything is about retinal parameters. And one of the most noticeable things about spiders is the variation in eye size and position. Obviously, these things impact field of view, sensitivity, and resolution, and both absolute and relative size do vary substantially. If you kind of just very, very roughly plot out their relative size, you can see quite quickly that our visual hunting groups do have more variation between eyes and within eye types as well. So there's definitely more differential investment. But obviously, position and orientation are also very important to field of view. So basically what space the eye is sampling in particular. So we know that spiders have quite different fields of view for different eye pairs. This is some really classic work from Mike Land, showing that you do see kind of overlap not only between eyes within eye pairs, but sometimes between eye pairs as well. So what we did to examine this was to take a geometric morphometrics approach. So each eye was given 2 landmarks at the center of the inner and outer surfaces and 11 sliding semi-landmarks around the edge of the eye. Basically, this means that you can register them and put them into, after kind of a Procrustes alignment, you can then essentially compare the coordinates of those landmarks in 3 dimensions between species as though they were multivariate data. So the 2 landmarks in blue and in pink there would be essentially homologous between the species we're comparing. This basically means that we can then analyze them via PCA to look at the relationships in a reduced number of dimensions, but capturing most of the variation in shape. So here they are colored by hunting mode, first of all, and you can see that there is actually some separation between the visual hunters and the other 2 groups. There's quite a lot of overlap between our web hunters and our free hunters, which is interesting. And there's less variation in our web hunters as well. And though, given that they've got a more similar lifestyle, maybe you could kind of extrapolate some meaning from that. And actually looking at a couple of examples of those species, we do have kind of a couple of interesting things coming up. So on the right-hand side, we've got our two visual hunters, which actually ostensibly look like they're shaped quite differently. And our two web hunters at the top left there, which look very similar, but occupy seemingly slightly different parts of space. However, obviously, as I said, phylogeny is a very important part of this as well. So if we superimpose the phylogeny onto our PCA, we can actually see that there may well be some confounding effects of phylogeny here, particularly in our visual hunters. But what you can do is then incorporate the phylogeny into your PCA. So basically, this is just making sure that the phylogeny is mostly explained by the first principal component. So if you look at our second principal component, which should actually relate only to shape, we still see this really nice separation between our visual hunters and our non-visual hunters. So now we can be fairly confident that there is probably some real signal here, perhaps relating to ecology. So what does that actually mean for eye configuration? So if we look at the maxima and minima of different principal components, we can start to unpick what those different components and how they change. So for example, if we look at PC2, so where we saw that kind of nice separation, we can actually see that there's a difference in clustering. So if you look at our wolf spider at the bottom right here, you can see that actually 3 of the eyes are clustered towards the anterior of the head. And in blue, which is our kind of maximum for that principal component, we see these 3 eyes clustered towards the front. But if we look at a web hunter here, we've got this very classic kind of halo arrangement of the eyes, giving a 360-degree field of view around the head with very little overlap. And that's what we can see at the minimum of PC2. You can see that the lines in the center here indicate orientation of the lens. So we can also then apply ancestral state reconstruction to this. Now that we've got the principal components and we have phylogeny as a factor, we can actually project backwards in time and start to look at how those principal components have changed. So then that obviously means we can look at correlations with ecological shifts and start to kind of piece together an idea of what ancestral spider visual systems might have looked like. So the bottom right here is just the prediction for PC2, so the component we just looked at in the most recent common ancestor of all spiders. And you can see that it does have this kind of distributed nature. And from this, we find that, as I said, the ancestral state was this kind of distribution of eyes. And actually the orb web spiders with this kind of halo arrangement kind of coincide, but then actually you also see this shift to anterior concentration of eyes in the same code as you see visual hunting. So obviously this is correlative. I can't pinpoint it and say that this is exactly the reason that you see this anterior clustering, but it makes logical sense, and it really it matches up very well with these kind of shifts that we see in spider ecology and evolution as well. So we kind of see 2 very different approaches then to having many eyes in spiders. And obviously, depending on the importance of vision, there seems to be 2 obviously equally good strategies to having many eyes. So at the one end of the spectrum, we still have our kind of relatively few types with not that much kind of divergence between the secondary eyes and the principal eyes. We also see a much more equal size balance, smaller maximum size of the eyes. And again, this kind of field of view is split equally between the 4 eye pairs. We do actually see some link again to this, not necessarily a sessile lifestyle. We don't have any immobile spiders, but certainly, in more plesiomorphic lineages, you might have kind of burrow hunting or sitting in orb webs for orb weavers. But actually, the kind of more active lifestyles, those visual hunting spiders and those ground hunting spiders, we do see greater divergence between eyes and eye types and more variable size, as well as this kind of anterior concentration and resampling of anterior space. So how exactly do these changes occur? Obviously, this is a huge question and deserves its entire own talk. But I just wanted to kind of take a closer look at some of the key features of eye configurations in spiders. So the really nice place to start here, of course, is eye size. It obviously has direct links to resolution and to contrast sensitivity, as well as being a good indicator of investment. And it's also, frankly, much easier to measure than many of the other characters that you might be interested in. So over the next few slides, I'm actually going to show you some work that was done by my postdoc, Luis Baudo and Gonzalez, looking at how eye size is patterned in developing spiders of different families and different visual ecologies. So he's actually been working over the last 1.5 years, I guess, nearly 2 years, to work with embryos of all these different spider species and start to look at what genes might be impacting how their visual systems develop. And as you can see, they actually represent a really nice range of different arrangements and sizes. So we have a good understanding of the genetic factors controlling eye development in Drosophila, of course, and because we have probable homology between the principal eyes of spiders and the ocelli of insects, and the secondary eyes of spiders and the compound eyes of insects, we can use this as a kind of blueprint to start thinking about what genes might be important. And there's been a small body of work on this before we got started, but what we kind of broadly taken from that is that we do see quite a lot of similarity between so Parasiatoda, which is this model spider, and with Drosophila. But we also find that, generally speaking, very similar retinal determination genes are probably expressed in the compound eyes and secondary eyes, and in the ocelli and principal eyes, which is very neat. One thing that we do see is that actually both of those eye primordia are probably surrounded and constrained by Wnt signaling. So that may well have quite an important role to play when thinking about size and how it's determined. One of the interesting things that, just because it's a vision audience, and I know that somebody will ask about it otherwise, is where's PAX6? Why do we not see PAX6 here as our wonderful master regulator? Many people before me and Luis have looked for PAX6 in developing spider eyes. In Parasiatoda, it just isn't there. And actually, in the other species that I just showed you a list of, we're not finding any real evidence of strong PAX6 expression in any of these eye primordia, which is an interesting spider weirdness. But there was a paper that came out only a month ago or 2 months ago from Natasha Tureczak and her group, demonstrating that they found expression of PAX2 in the areas that you might expect PAX6 to actually to appear instead. So there's a really interesting potential story there about co-option of PAX2 to do PAX6's job. But that's an aside. I won't talk about it today, but I thought you would be interested to hear it. Go and read their paper. So one of the other great things about spiders that really gets people kind of interested in the genetics and the development is that along with other arachnopulmonates, they've actually undergone a whole genome duplication. So we see a lot of the genes that, although they're possibly doing the same job as in Drosophila, a lot of them are present in duplicate in the retinal determination gene networks. However, although this seemed like a very tantalizing opportunity to then sub-functionalize and use that as a toolkit to make your eye configurations very diverse and very crazy, we actually don't see that much difference in copy numbers between any of these genes, between any of our families. So, it's not necessarily about what these genes are doing. And in terms of sequences, they're also not very divergent. So probably isn't down to this whole genome duplication and the abundance of possible determination genes. But that must mean that it's probably about how these genes are expressed. So this project started in August 2020, so in the kind of like depths of lockdown. It's mostly been a COVID project, but it means that Luis and I have spent the last year or so collecting spiders and raising colonies of spiders in our houses, at least at my end, much to the chagrin of my husband. But it means that we have been kind of trying to keep these things alive at home to some extent. And of course, the purpose of this is then to get embryos, these kind of like very cute jelly baby versions of spiders, for in situ hybridizations to look at where these genes are expressed. So I'm just going to show you a couple of these different genes, and I realize this is a very busy, overwhelming slide. Don't worry, I will pick out what is important. Just a couple of the things that we've noticed from some key genes. So first of all, we were looking at Sumner-Rooney, which is one of these kind of great marker genes for looking at where the eye primordia are. And if we just look at these three species, so we have Pardosa amentata on the left, which is a wolf spider, Malpissa muscosa in the middle, which is a jumping spider, and Volcus phalangoides on the right-hand side, which is the daddy long-legged spider, the one that will sit in the corner ceiling of your bedroom and spin wildly if you bother it. So one of the things that we can see here is that in both our wolf spider and in our jumping spider, we do have different sized primordia in a kind of coarsest sense for the different eye pairs, and they do broadly reflect adult eye size. What we don't see is the same thing in Volcus. I think is kind of an interesting little comparison. The other thing is that now that we've looked at Sumner-Rooney's expression in these two species that have very large eyes, something that wasn't previously visible from examinations of Parasiatoda, which has quite small eyes, is that it also looks like there's this kind of two distinct areas. So there's this halo of expression, and there's potentially a more kind of concentrated dot here at the ventral side of the eye. So that could be an interesting tidbit and a good lead to follow up on maybe. We actually see something very similar when you look at eyes absent. So if you actually again look at those same three species, we have really nice demonstration of different eye sizes in these two species. But again, Fulcus has these kind of, they're clearly distinct, three different primordia for the secondary eyes, but they're a very similar size to our principal eyes here, when actually in adults, they're much, much larger. Finally, just one more because I think it's interesting and I kind of want to hear people's thoughts on it, but it remains somewhat mysterious. We also looked at 6.3, so this is optics in Drosophila. This is only expressed in the secondary eye pairs, but I just wanted to kind of draw your attention to this expression pattern in Marcissa. So this is one of the cases where we do have two copies. Both copies are expressed in the eyes, but they seem to have very different expression patterns, which I think is quite interesting. So you can see that the one copy on the left-hand side here is only a very small concentrated dot that again does not reflect eye size. But if you compare that with the autolog on this side, the expression patterns of 6.3.2 almost look like they wrap around those expression domains of 6.3.1. Again, this particular gene does reflect differences in adult eye size, but its counterpart does not. So this is one I'm keen to follow up on, but would be very interested to hear interpretations of those expression patterns. So if developmental patterning can't explain everything that we're seeing varying in adults, then there might be something else happening during growth as opposed to during development. So this is some work from Kaylin Chong, who's an undergraduate working in my lab, and she looked at static allometry in a range of different species with different visual ecologies. So allometry is where if you have a feature that grows at a different rate from the rest of the body. So the classic example is the human head, which exhibits very strong negative allometry. You have giant-headed babies and small-headed adults, which often reflects kind of a greater necessity for investment early in life. So Kaylin actually measured some crazy number of specimens, I think more than 1,100 specimens from our collections in Oxford, across 36 different species and 6 different families with variable visual ecology. So she actually just took a carapace width measurement and then diameter measurements of those 4 eye pairs. And actually, if you do have a look at, we haven't processed all of these data yet, so I'm just giving a kind of quick overview of this. But if you, for example, compare salticids, jumping spiders, and araneids, which are orb weavers, a visual and a non-visual family, there's a clear difference in kind of size and distribution. So obviously that's just the height of the slope, but also possibly in growth as well, so the gradient of that slope there. For comparison, there is some other work on eye allometry in spiders. So this really nice paper, again from, I think from Nate's lab, showed really nicely that you do get negative allometry in salticid eyes. This was looking at developmental stages, or ontogenetic stages, sorry, so different instars across that initial growth. So negative allometry here makes a lot of sense. Obviously, vision is crucial for survival in these animals, so they invest super early. What was really interesting that we found in our adults of different sizes, that actually we found isometry in all the eye pairs that we looked at, in particularly these 2 salticid species. So this basically means that the slope is not significantly different from one. The eyes are just growing at the same rate as the rest of the body. So it's interesting for a couple of reasons. So, there could be a shift in growth dynamics perhaps, but it's interesting because we were expecting to just see a continuation of this negative allometry that Nate's group had demonstrated really nicely. What we did see in what's interesting, we did actually then see that in our non-visual group in the araneids. So here we did find isometry, again, the principal eyes, but the 3 secondary eye pairs all showing that kind of negative allometric growth. So not sure whether to interpret that as, there's continuing investment, from salticids in those eye pairs as they grow. Positive allometry is not that common, but it's seen in things like sexual selection characteristics. So it could be that it's just a very important trait. They kind of keep investing as the animal grows and grows. If you just look at the salticids and you kind of make a comparison across different species, you can also see that there's a clear difference between eye pairs as well. So even though we saw that all 4 eye pairs exhibit isometry, there's clearly something slightly different happening in the different eye pairs once you look across different species. So there's a really, really tight relationship there between body size and eye size in the principal eyes, but it seems to be kind of much looser once you're looking at the posterior and median eyes, which are broadly thought to be probably vestigial and not doing very much. If we come back to our falsehoods, though, we still don't have a satisfactory answer as to when the size of those secondary eyes is established. So what we actually see here is that the secondary eyes are negatively allometric again. So this should mean that they have already invested in that eye size and they don't grow any further. So this does contradict what we've seen in our gene expression studies so far, because we didn't see this kind of like reflection of adult eye size. So either we're missing something, so there could be something happening in those instar phases where the Gautier paper showed that in salticids. Alternatively, it could be that there are other genes that determine and pattern eye size in Volcus that we haven't tried in our institutes yet. So if we come kind of back round now, I suppose, to our overall like proposed spectrum, we kind of wanted to suggest that there was this range of different approaches to having many eyes, with a high eye number, few types, and maybe a kind of sessile lifestyle at one end, and lower eye number and larger or more specialized eyes at the other end. And we do see generally that that is quite well represented within spiders as well. They cover that kind of bite-sized part of that overall spectrum, but those overall patterns kind of follow the same vein. And that's removing, variables like broader kind of phylogenetic constraint. It's removing eye number from consideration. So they do actually give us, hopefully, quite a nice closed system that looks like it might be representative of that whole spectrum, which gives us a really nice opportunity then to explore some of the questions that we've set out today. And one of the particularly kind of interesting bits is that the possibility that we're seeing a transition from duplicated systems to parallel systems in some of those secondary eyes there. So of course they still share their developmental origins, but it could, we're seeing them try to collect different information despite that kind of shared origin. So there's also the opportunity to then try and add in variation in eye number in future and in the number of eye types. So for example, particularly in this more plesiomorphic part of the tree at the top here, we actually do see quite a lot of variation in some families in eye number. So I've just spoken about families that have 8 eyes today. Actually in several different groups, particularly the caponids are a really interesting family for this reason, is that they have species with 8, 6, 4, 2, and 0 eyes, which is really interesting. So obviously, where you do have species that lack eyes completely, there's usually a very obvious ecological reason. For example, they might inhabit caves. But for the caponids, you get all these in-between combinations as well, which I think is really, really interesting. So something that I want to kind of do a bit further down the line is looking at whether we then see compensation, for example, for loss of part of the field of view being covered. Do we see divergence between the remaining types, particularly where the principal eyes are lost? So these families are possibly a really nice new resource for kind of further information on the importance of eye number specifically. So that's as far as I kind of wanted to take you with the spiders and particularly with the overall survey as well. But before I hand back over to George, hopefully take some of your questions, I'm going to shamelessly plug a couple of things that we have going on. So first of all, and for me, most importantly, we're currently hiring. So I'm looking for both a postdoc and a PhD student to start in the next couple of months, the next few months. So if you or anybody you know or anybody in your lab is looking for a position, please encourage them to check out the website or get in touch with me if they have any questions. Both of those positions will be closing in the next few weeks. But again, do get in touch with me if you have anybody in mind or if you want to know more about either of those. And secondly, if you're interested in the kind of like broader concepts of these multi-eyed visual systems, there will actually be a book coming out, which has been edited by Elka and Mike as part of the Springer series in vision research. So hopefully that will be coming out at some point in the next year, perhaps, but that will be, it's a compilation of work by many, many other people. But if you're really kind of want to get your teeth into more of these kind of weird and wonderful systems, that'd be a very good place to do it. And that just brings me to my last slide, so to thank everybody who's been involved in this work at every level, from collecting specimens and delivering things to working in the group, and of course to thank my funders and to thank you all for your attention as well.
Thank you very much, Lauren, for this fantastic talk. the complexity of the visual systems out there is like amusing, captivating, and of course alarming, because if we only focus on model groups, then we miss or misinterpret so much information. So for the time being, I will try to just act as a moderator and convey questions that appear in the chat. I have already posted the Zoom room link in case someone already would like to join us. And the first question appearing is from Madine Sarvestani. Do we know whether spider head movements preferentially stabilize gaze on principal eyes or all eyes?
That's a great question. So actually, one of the really nice things that I didn't mention about spiders is that they the retinas have muscles to move them around as well. So actually, they wouldn't necessarily need to use head movements for gaze stabilization. They actually have the ability to move the retina around the back of the lens of the eye itself. So I know that looking at gaze while static, a lot of work has been done with eye tracking by a couple of the groups here in the US. While moving, I'm not sure actually. I would assume that there probably is some way of stabilizing the gaze while spiders walk. Anecdotally, from watching visual hunters hunt, they tend to stop and watch rather than kind of move and track prey. But that's something that a jumping spider expert would have to weigh in on, I think.
Right, and down this topic, so do you think that different spiders use vision more than others for species recognition, like trying to draw parallels with what mantis shrimp visual complexity conveys as an advantage? Do you think like different spiders would rely on a different extent for species recognition in their vision?
Absolutely, yeah. So we can certainly see that, I mean, they're not the only example, but the jumping spiders are definitely the best example of this, where not only for species recognition, but, you know, there have been really beautiful studies looking at how individuals track different, you know, body parts of another individual during, for example, a mating display. So certainly there's ability for not only your own species recognition, but I think also for there's been some nice work looking at the comparison of silhouettes of possible prey items with other silhouettes. So certainly where the resolution is high enough, I think there is definitely some recognition going on there. Obviously, if you're a web hunting spider, the reliance on vision is very, very low. But we do, you know, obviously in a kind of trade-off with things like vibration sensitivity, there you do have not necessarily species recognition, but very good recognition of the different signatures of vibrations of different possible prey items as well. So definitely differences across different spider species for sure.
Right. Thank you very much for that. So next question is from a user named Mathenol. Have there been studies which either blind or inhibit input from specific eyes and look at subsequent behavioral changes?
Yes, yeah, absolutely. So again, this has mostly been, but not exclusively, in jumping spiders. There was there's already very well established work that means that the anterior lateral eyes and the principal eyes, so these kind of pointing in the same direction, that covering one set means that you kind of lose that recognition of movement, and covering the other set reduces the amount of detail that can be seen. So there is some really nice work explicitly demonstrating that, you know, those two eye pairs facing the same direction are doing very, very different jobs.
Right, and given that you mentioned the jumping spiders, so like I think there was a paper maybe like around 10 years ago that they examined the jumping spiders, and they found that they have like two photoreceptor layers, one having the image in focus and one out of focus, and this would help with depth perception, like within a single eye. How specific is this to jumping spider? Like this kind of complexity, is it evident across the spider tree or?
So in terms of kind of multi-layered retina, I mean, it's definitely greatest as far as we know in the jumping spiders, but in some crab spiders, for example, in the principal eyes, you do also get a kind of a slightly elongated two-level retina there as well. So right. There are examples of being elsewhere, but it's one of those things where jumping spiders are the kind of Olympians of visions. They've received a lot of attention, a lot of behavioral work. They're also you know, the best to get a behavioral reaction out of. Mm-hmm. There are many spiders that you can't get to, you know, surprise them with a lube. They might not react, but they've been a really exceptional model. But certainly from morphology, we can see similar trends in some other groups, but it's not really to the same scale as it is in the solstices. Right.
And before I convey the next question that appears, I would like to let you know that there were both greeting messages at the beginning and thank you messages at this point. So, you know, because you don't have it open, like it's not that it's just the two of us in the void. So the next one appearing in the chat is from Helder Hugo. In Salticidae, sexual dimorphism can be quite evident. Did you consider looking at the differences in eye or vision between males and females?
So we actually basically tried to only use mature females for these structural studies. So absolutely. There's obviously a really important component of sexual communication, particularly in those groups, but possibly also in groups that we don't know about or haven't studied quite as well. We thought that the safest thing to do would be to stick to just one sex and to just use mature females. So because at least at the moment our focus is on kind of comparing between these families, we've tried to keep that consistent. But absolutely there may well be differences that we see between males and females. We haven't looked into that just yet.
Right. Thank you very much for that. And the last question appearing in the chat, and this would be the last that I would ask you to address indirectly. People are more than welcome to join us in the Zoom room, for which the link I have reposted. So the last one appearing is from Sam Budov. Thank you. Super interesting talk. With respect to the topographic asymmetries, are there retinal specializations like streaks or area centralis? What are the topographies like between eyes in a single species?
So the kind of high resolution reconstructions of the retinal mosaic are still ongoing. So we've only got a couple of species for this so far. We are definitely seeing, at least in the species that we've got, so these crab spiders that we're looking at, there does seem to be a kind of horizontal streak along the middle of higher resolution, kind of reduced skew as well. But one thing we're seeing is also kind of sometimes some kind of almost like a disruption, like a vertical line down the center of the retina. Whether this is, it could be a structural artifact, whether that's where the photoreceptors are then being kind of carried through the tapetum is not entirely evident yet, but there are definitely structural features in the retina that keep coming up. So this horizontal streak is definitely one of them in the visual hunters.
Right. And before I terminate the broadcast down this path, so you saw, like, for example, the area of high density for photoreceptors, or areas that the rhabdomeres are of different size. And do you think like this would reflect also what happens like for the downstream layers?
That's a great question. That's something I can't give you a satisfactory answer for right now. I mean, there's some really nice work by Sky Long and some of her colleagues on the visual neuropils and kind of how the morphology of the visual neuropils varies between groups with different reliances on vision. And it's a really quite staggering difference. It's the point of having more or fewer visual neuropils in different families, which is pretty, that's a pretty large scale evolutionary change to your brain structure. So I'm certain that there'll be a reflection of more detailed points as well. Absolutely.
Right. So thank you very much once again, Lauren, for this fantastic talk and so many organisms and like the complexities like outwardly. So I would like to thank the audience for being here, and I would be terminating the broadcast. The Zoom room link is still visible, so in case you would like to keep track or participate in the ongoing discussion, please make sure to click on it. Thank you very much.
Thank you very much.
And we are officially offline, and people are already here.